Very welcome to this YouTube channel. I'm Usha Yadav, and today's lesson is about your lectin pathway of complement activation. All you know that we had already completed the another mode of complement system activation, in which we studied about the classical pathway and the alternate pathway of complement activation. So in this particular video lesson, we are going to discuss about the lectin pathway, which is based on interaction with the carbohydrate moieties that present on the surface of any pathogen or the invading bacteria, right? So this particular video lesson is about the complement system activation, but the third mode of activation. Okay, we had already discussed about the another two. You can go through the previous video and you can understand the basic concept of the classical pathway as well as the alternate pathway. Okay guys, so let's start with the lectin pathway. So if we talk about the basic of lectin pathway, then lectin pathway, its name is showing that what kind of function is going to perform this pathway. Okay, lectin pathway, all we know that this pathway is going to used for the activation of the complement system, but lectin, its name is showing that somehow it is going to interact with the carbohydrate moieties, because lectin is a kind of protein which show its binding with the carbohydrate moiety, specifically the mannose. Okay, and these mannose or the carbohydrate moieties are present on the pathogens or the invading pathogen. For example, we can say that on the bacterial membrane, because all the plasma membrane, as they carry phospholipids, all they also carry on this on their surface some some many of carbohydrate moieties, and these carbohydrate moieties are responsible for their recognition by the mannose binding lectin pathway. Okay, because its name is showing that mannose binding lectin, it means that the lectin pathway is going to bind with the mannose. It's showing its binding with the mannose carbohydrate moiety, right? So another name of lectin pathway is mannose binding lectin pathway, or we can say that mannose binding lectin pathway or lectin pathway simply. So lectin are carbohydrate binding protein because they specifically bind with the mannose, which is a carbohydrate moiety. So it is a carbohydrate binding protein, and these carbohydrate binding protein are present in our cell body, so normal जो हमारी immune cells होती हैं उनके surface के ऊपर, but the carbohydrate moiety through which they are showing their interaction are present on the pathogen. Okay, so the lectin pathway, like the alternate pathway, does not depend on antibody for its activation. If you memorize that, when we discuss about the classical pathway and the alternate pathway, then we discuss about the initiating point and the execution portion of the pathway, particular pathway. So if we talk about the classical pathway, then the initiator point or the initiating protein activation is dependent on antibody antigen interaction. Okay, on the other hand, if we talk about the alternate pathway, then there is no the recognition process is not dependent on the antigen antibody complex, but it directly depend upon the antibody. But if we talk about the lectin pathway, then lectin pathway is not dependent on antigen antibody complex, as well as it does not depend upon the antibody. It just bind to the mannose that present on the particular pathogen. Okay, somehow it's a mechanism, or it's a processing is like your classical pathway, because its protein, the initiator protein of the lectin pathway, also act just like the initiator protein of the classical pathway, but because these proteins are different, quite different, but their functions are quite similar, that's why we just compare the classical pathway and the lectin pathway, because the through the complete process of this complement activation in classical pathway and lectin pathway is quite similar. Okay, so activated by binding of mannose binding lectin to mannose residue on glycoprotein or carbohydrate on surface of microorganism, mannose binding lectin bind to the surface of cell or pathogen. As I already told to you, okay, yes, the mannose binding lectin bind to the carbohydrate moieties, and there is so many proteases are associated with this MBL, in which we generally include the MSP1 and the MSP2, because as the mannose binding lectin get bind to the get bind to the mannose that present on the pathogen surface, get interacted, the recognition process is completed, and after their recognition it will activate the another protein, okay, the other another signaling cascade protein like MASP1 and an MASP2, because these proteins show their proteases activity, and all you know that proteases are those enzymes which conduct the cleavage of the protein molecule, okay. So the MASP1 and 2 protein have structural similarity to C1R and C1S and mimic their activity. Just like that, your classical pathway, in your classical pathway, C1Q has the capability that it can recognize the invading pathogen. MBL can recognize, the recognition process is done by the MBL, and after recognition, C1R and C1S are those protein which are going to activate the C3 convertase, you know, the complex forming component, the C4 protein and the C2 protein, hai na? Just like the same way as the classical pathway, activation is conducted by these proteins. Lectin binding pathway is also conducted, or we can say that activated by the activation of this MASP1 and MASP2 protein, because MASP1 and MASP2 protein have structure similarity just like your C1R and C1S, so that they also mimic their functionality. The active complex formed by the dissociation, but the association cause cleavage and activation of C4 and C2, just like your classical pathway. As this MASP1 and MASP2 are activated with the help of this mannose binding lectin protein, then in turn, after their activation, they in turn activate the C4 and C2 and form a complex, that is C3 convertase, which is further going to cleave the C3 protein into two unit, C3A and C3B. And all you know that C3E is showing your inflammation response, and C3B is going to forming another complex which is a term as C5 convertase, and the C5 convertase is going to cleave the C5 protein, and then again the MAC complex formation, okay. So this is an image which is showing the complete lectin binding pathway, or the overall mechanism of complement system activation via the lectin pathway. You can see that in lectin pathway, the initiating point or the initiating proteins are MBL protein and the MASP1 and MSP2. After that, the complete process is just like your classical pathway, okay, but the initiator point is quite different, but the activity of those proteins are quite similar, just like your C1R and C1S protein. So if you start from here, then you can see that mannose binding lectin bind foreign surface, because all you know that the mannose binding lectin show the direct interaction with the pathogen by show its binding with the mannose that present on the surface of the pathogen, okay. There is no antibody interaction is there, so you can see that the mannose binding lectin is directly going to interact with the mannose carbohydrate moieties present on the pathogen surface, and after interacting with that carbohydrate moieties, it is forming a complex, or we can say that it will initiate the proper signaling cascade of complement activation, okay. So mannose binding lectin bind foreign surface directly, and it will initiate the signaling cascade of the lectin pathway. You can see that the mannose binding lectin shows binding with the mannose carbohydrate moiety, and after their binding it will recognize the pathogen, or we can say that any bacteria or any viral component. As it recognize its next step is to response, okay, because all you know that in immunology there is a two-step process is conducted by the immune cells or the complement system. First one is recognition and next one is a response. So recognition is conducted by the NBL and MASP1 and MSP2 protein in case of lectin binding pathway, and in case of classical pathway it is conducted by the C1 protein which has 3 subunit, and in case of alternate pathway it is dependent upon the C3 protein, okay. So if we are talking about the lectin pathway, so just focus here that first one is binding. After that it will cleave the C4 protein into two components, C4a and C4b, and C4a is going to release in the blood serum, and C4b is going to form the complex that will result in the formation of C3 convertase with the another protein that is known as C2. C2 protein is again going to cleave into two parts, C2a and C2b, in which C2b is released in the blood serum and C2a is going to form the C3 convertase complex. Okay, so after the formation of this complex, you can see that here is the complex which is going to act as a C3 convertase enzyme, and it shows the enzymatic activity, and this C3 convertase is going to cleave the C3 protein into two units, C3a and C3b. Again the C3a is going to release in the blood serum, and C3b is going to form the complex which will result in the formation of C5 convertase, and this C5 convertase is going to cleave the C5 protein. As a C5 protein is cleaved into two units, then it will activate the MAC complex or the membrane attacking complex on the pathogen membrane. And all you know that this MEC is going to form a pore like structure in the membrane. As its pore like structure is going to form, the intracellular material or the intracellular matrix is going to move toward the outside, and the pathogen is going to disrupt. Okay, so this is your lectin binding pathway, in which you can see that it is quite working just like your classical pathway. Okay, another is that if we can see that lectin pathway is directly or indirectly is going to initiate the classical pathway and the alternate pathway. So this is the pathway in which it is going to activate the classical pathway, and it also can act to activate the alternate pathway. How it will going to activate? After the C3 convertase formation, it will interact with the factor B and factor D and properdine protein. Okay, and after the formation of the C5 convertase, again the same process is going to take place. Okay, but the process that which path is going to followed by this lectin binding pathway is dependent on the type of protein that are going to used in the formation of C3 and C5 convertase enzyme. Okay, so overall if we discuss about the overall mechanism of complement system activation, then we can see that there is three pathway: alternate pathway, classical pathway, and lectin pathway. Alternate pathway is antibody antigen independent pathway, in which there is no complex formation is there, and it can be activated with the help of C3 enzyme, because in this pathway you can see that there is a there is a multi-step process, but there is two position where the C3 protein is going to use. But the difference is that the first C3 protein is hydrolyzed, okay, and the next protein is without any water molecule. So the C3 protein is going to act as an initiator point, and here is the C3 protein is going to the executor because it will further going to activate the C5 protein, and C5 protein is going to recruit the MAC, okay, membrane attacking complex. If you talk about the classical pathway, then you can see that here is the initiator point is the C1, or the recognition process is done by the C1 protein. C1 protein has three component unit: C1q, C1r, and C1s. Only the C1q unit has the capability that it can recognize the pathogen. After its recognition process is done, it will further activate the C1r and C1s, and C1s has the capability to start the signaling cascade to activate the classical pathway, in which you can see that again the C3 convertase is formed, and C3 convertase again form the C5 convertase, which will further recruit the MAC, okay, membrane attacking complex. Lectin pathway, we already discussed about it, that there is MBL and MASP proteins are present, which are also going to act as like your C1QRS unit, and as the recognition process is done, they are going to form a C3 convertase enzyme, and then this C3 convertase enzyme is going to cleave the C3 protein, which will further form the C5 convertase. C5 convertase again cleave the C5 protein and will result in the recruit or we can say that in the functionality of the MAC, Okay, so this is overall about your this is all about your complement system activation. I hope you guys understand this lesson properly, and if you guys face any difficulty in understanding any concept regarding this topic, then you can ask me in the comment section. I will try my best to resolve your all of doubt, or you can